Okay, today's talk is the influence of water withdrawals on spatial distributions and population numbers of species in Charlotte Harbor. I'm a research scientist with Florida Fish and Wildlife. For about the last 23 years, I've been involved in developing habitat suitability models within the Center for Spatial Analysis. Christy Santi is a GIS analyst that's worked closely with me and prepared many of the maps that I can show today. Dr. Chen is a hydrodynamic modeler with SwiftMud. And so, you know, I think this study is an important collaboration between Florida Fish and Wildlife and the Southwest Florida Water Management District. So we're bringing together hydrodynamic modeling with the monitoring that's been done by my agency. Jonas Gile is the program manager for my project and for a number of other projects that have to do with freshwater inflow studies and in rivers within the district. And so that's where we stand with that. And next slide. Okay, so the goal of this particular study, which started in about 2014, was assess the influence of environmental conditions on fish and invertebrate species to support setting minimum flows and levels for the Lower Peace River and Charlotte Harbor. So, as most of you probably know, there's water withdrawals that occur at the head of the Lower Peace, the tidal portion of the Lower Peace River, and also on the adjoining Shell Creek that joins the Lower Peace River. There's also a water withdrawal site, I think, near Punta Gorda. So the objectives of this study were to conduct habitat suitability modeling, which I'm calling HSM, and habitat mapping to seasonally assess spatial distributions and population abundance of selected species life stages. So during phase one of this study, I just used our fisheries independent monitoring data, or FIM data as we call it, to run these models, habitat suitability models, and map habitats from 1996 through 2013. So I did a baseline study here where we mapped habitats just using our FIM data, and we linked that with our habitat suitability models. And there is a publication that came out on that in 2019. But you know, that statistical analysis indicated that salinity was highly significant for most of the 8 species life stages that we were working with over 4 seasons. So that's like 32 species life stage analyses. And but you know, the main factor there was salinity is highly significant. Or I think somebody said, well, don't say highly, just call it significant. Okay. But, you know, the main, in 2017, I gave a presentation to this group about baseline work that we had done. So we're using FIM data to run habitat suitability models and outputs from hydrodynamic modeling done by Dr. Chen. So the hydrodynamic modeling allowed us to map temperature and salinity patterns seasonally from 2007 to 2014. So that's the focus of this present study, is that time period. And today I'm going to talk about the baseline, but I'm also talking about phase 3, where we did water withdrawals, or the water withdrawals are occurring. And again, we're using FIM data to run habitat suitability models and outputs from hydrodynamic model. But we're producing the temperature salinity maps for minimum flows. So we got baseline flows and minimum flows that we're analyzing and comparing in this present study with the goal of estimating population numbers. So the actual estimation of population numbers from habitat suitability maps, I think, is kind of unique. It's something that I've developed. It's not something that's generally available to. We have these long-term fisheries independent monitoring studies in estuaries, but in most cases, they don't analyze the data to estimate population numbers or map the spatial distributions of the species in estuaries. even with my own agency, but with other agencies as well. Next slide. Okay, so SwiftMud has developed a percent of flow approach using minimum flows to maintain 85% of the most sensitive salinity habitat, less than 2 PSUs, which I'm just going to call parts per thousand. So they're concerned about managing the salinity habitat in the oligohaline section of the upper portion of the lower Peace River, and I guess also, but I'm not talking about the lower Myakka River. So they want to compare, we want to compare baseline conditions in the uppermost part of the tidally influenced lower Peace River. This assumes that when seasonal salinity and temperature predictions do not differ substantially between baseline and minimum flows, the associated water withdrawals should not be detrimental to biological resources in the river and Charlotte Harbor. So I might mention that the percent of flow approach for many agencies is assumed. In other words, percent withdrawals, if you like, are used on a, they have to be done on a day-to-day basis. And so decisions about how much water to remove have to be made daily. The present study is using a, we're trying to assess population numbers. So we actually have, we want to know what the impact of water withdrawals are on biological resources in the study area, including the lower Peace River and Charlotte Harbor. Next slide. So this is a daily hydrograph for the period that I mentioned, 2007 through 2014. So the peaks that you're seeing here are daily, but I've got years across the bottom, so you can see that, you know, we have about 4 seasons of the year. The fall, the winter, and the spring are basically dry periods. There's some fluctuations there, some rainfall, but inflows are low. Then you get into the summer where you have a lot of rainfall, and so you get high inflows. So what the Water Management District is doing is withdrawing more water when they have higher inflows. So during the summer, the inflows, or the amount of water that's being taken out, can be as high as 40%. So what you see in this graph, though, is the you've got the baseline graph, and then on top of it, you've got the minimum flows graph. And so what you see is the difference between baseline and minimum flows in blue. That's the area. So what you see in blue is actually the amount of water being withdrawn in cubic feet per second, because it's the difference between baseline and minimum flows. So you can see how that works. And hopefully I explained that clearly, because I think there's some confusion with some people, including myself. But more recently, next slide, please. SwiftMud is now presently considering putting a cap on water withdrawals by limiting that difference between baseline and minimum flows. So here you can see that there's a much smaller amount of water being withdrawn because they put a 400 CFS limit on how much water can be removed. So I guess there was some concern, if you look at the previous graph, that they seem to be taking out a lot of water. The question in my mind is, well, even in the previous graph, what influence did that have on biological resources? So my study was focused on the previous graph, in the sense that this idea of a 400 CFS limit is a new idea that's coming out in the 2020 management plan by SwiftMud. But prior to that, in my study, did not take into account a 400 CFS limit. Next slide. So Dr. Chen, he's concerned about trying to predict salinity patterns in the uppermost part of the lower Peace River above where it joins Shell Creek. So the area in yellow at the top of the graph is what he was concerned about, predicting salinity in the so-called oligohaline zone. And so I said earlier that they're trying to manage water withdrawals so that it's less than 85%, for minimum flows compared to baseline. And so you're talking about managing salinities in that zone. But to do that, he's modeled it all the way down past the mouth of the Caloosahatchee. And he's got a hydrodynamic model that predicts salinity and temperature over this yearly from 2007 to 2014. And so it's a complicated model that has two components, a 3D component, a submodel, and a 2D submodel. And but it basically, it's, you know, we've taken, Florida Fish and Wildlife, our GIS group, obtained the data from Dr. Chen, and we've used that for modeling and mapping temperature and salinity patterns in Charlotte Harbor. So we're not concerned about this entire area, but we are concerned about the area north of Pine Island. Next slide. Okay, so when we did that, you can see here we have seasonal salinity maps that were produced by Christy Santee. And so you've got baseline flow salinity maps for fall, and then to the right of it is the baseline flow salinity map for winter, and then below that is spring and summer. And then when you look over to the right, you've got the same similar maps, very similar maps for salinity, but they're for minimum flows. So again, you have fall, winter, spring, and summer. But when you look at these maps, it's very hard to see a difference. So while there may be something going on in the upper part of the lower Peace River, it didn't seem to have much effect on Charlotte Harbor going all the way down to Pine Island. And so one of the findings, I thought this was something that Dr. Chen had engineered, but it turns out that this is something that was unpredicted from, nobody knew that this was going to happen. And so this is an original finding that, and it seems to be the case for all of the species life stages that we analyzed, that we're getting similar temperature and salinity maps for baseline compared to minimum flows. Next slide. So here you can see the temperature maps, again, for fall, winter, spring, and summer. On the left for baseline, on the right for minimum flows. And again, there's very little difference in these maps, but there's supposed to be a difference here. The modeling that we did was predicated on the idea that we're holding the other environmental variables for depth and bottom type and dissolved oxygen constant. So the difference that we're looking at here, and it was part of the analysis that we were planning to do, is comparing the differences between baseline and minimum flows for temperature and salinity. Next slide. Okay, so fisheries independent monitoring being done by the Fish and Wildlife Research Institute within FWC. There's actually about 4 kinds of seines of different sizes. And then there's a trawl that's also used in Charlotte Harbor. So we had to take the data that was collected on a variety of species and analyze it and try to standardize the catch per unit effort, or CPUEs, between the different gear types so that we could do our modeling. Next slide. So the species that we analyzed in the present study included juvenile plus adult hog choker, juvenile sand sea trout, juvenile plus adult blue crab, early juvenile southern kingfish, adult bay anchovy. We've actually also analyzed juvenile bay anchovy. And then we have studies for those are, so anyways, the species I just listed. Hello? Hello? You'll have to excuse me, I'm getting crank phone calls. Okay, the estuarine residents are the species that spend the majority of their time in the estuary. Estuarine and reproduce in the estuary. Estuarine transits like early juvenile red drum and early juvenile spot reproduce in the Gulf of Mexico, and then the juveniles and early juveniles and juveniles move into the estuary. So they're still estuarine dependent, but they're not reproducing in the estuary. Next slide. So what I'm calling a habitat suitability modeling is actually what we've done is use delta gamma generalized additive models, so delta GAMs if you like. And so that the program we're using running in R developed actually out of 5 factors, there's 31 combinations of 1 to 5 factors. So it actually developed 31 different models, and then so it's a submodel that fits splines across of abundance or CPUEs across environmental gradients for positive catch rates, and then frequency of occurrence, which is basically looking at the percentage of zeros in the data sets. So we had 2 submodels, and then the splines were combined to produce predicted gear-corrected CPUEs, so gear-corrected abundance, if you like. And so the final models that were chosen were based on having the lowest Akaki information criterion, or the lowest AIC, to choose the best delta gamma GAM. The best GAM developed was linked to interpolated habitat grids. So the habitat grids, which are primarily the ones that we're concerned about at temperature and salinity, have about 1.9 million 15 by 50 meter cells. So what we're trying to do is transfer the abundance values off of the fitted splines to the habitat grids. So now we have, for each habitat grid, a grid with gear-corrected CPUEs, a continuous grid of CPUEs. And then those CPUEs were averaged across the different habitat grids. And then we had to divide the continuous CPUE grid that was produced into 4 zones using Jenks natural breaks. So we're clumping the data from low to high abundance, if you like, based on the differences in the grouping of the low to high abundance zones. So it's a way of breaking up your data, continuous data, into zones, so that we can produce habitat suitability maps. Next slide. So this is an example of some of the fitted splines after the positive CPUEs and the frequency of occurrence splines were combined. This is a gear-corrected CPUE splines and histograms for juvenile to adult hog choker based on the full model. You can see the in terms of salinity up on the top left-hand graph that there's a marked preference for low salinities, less than 5 parts per thousand. This is done in the spring, but they seem to be abundant at about 10 to 20 parts per thousand. If you look at the dissolved oxygen and depth lines, they don't look like they're significant, and the statistical analysis in most cases did not find them to be significant, but in some cases they were. And so for some species models, dissolved oxygen and depth are included. Also the same for bottom type. But we have a year term, and we have our categorical variables for bottom type, year, and year type also included in the model. Next slide. Okay, so what I'm comparing here is I just wanted to look more closely at these hog choker, the predicted habitat suitability maps for hog choker, and the blue zone at the top of the lower Peace River represents the optimum zone, of the zone of highest mean abundance. And so there's 4 zones in here. The yellow is the low zone, the orange is the moderate zone, the green is the high zone, and the blue is the optimum zone with the highest mean CPUEs. But if you look again, you're looking at your habitat suitability maps. So at the top you have baseline analysis, and at the bottom of the graph you have the minimum flows. So again, you don't see much differences between the baseline maps for each season and the minimum flows maps for each season. They're almost identical, but there are some subtle differences, and we wanted to try and figure out how much of a difference is there. I mean, we deliberately chose baseline and minimum flows because with these factors, especially temperature and salinity, because we wanted to see the differences. But the maps don't show much difference for the habitat suitability modeling maps. Next slide. Here we have the juvenile to adult blue crab maps. There's well, it looks like they're reproducing in the estuary in the winter, which is on the top right. And then those abundant zones, the blue zones, contract, and then they expand again in summer. So it's kind of interesting that there's 2 seasons where you have a rather high abundance of and rather broad distribution of the optimum zones in blue, which is the winter and summer maps. And so part of the explanation here is that they seem to be recruiting at those 2 seasons of the year. But you can see in the bottom map for the summer that there is some expansion of the optimum zone down into northern Charlotte Harbor. Next slide. Here you can see a better case of, I think it was a general case to say that when you have a lot of rainfall in the summer, you get an expansion of the geographic range, particularly the optimum zone expands down into Charlotte Harbor. So you can see that in the summer map for adult bay anchovy. The interesting thing about all of these seasons, though, is previous work I did in 2019 indicated that each of these species life stages have a preferred salinity range that stays about the same between seasons. So the salinity range they prefer stays about the same, but the geographic range of that, particularly you can see it with the optimum zones in this, expands as so if let's say they prefer 10 to 20 parts per thousand salinity, that if that 10 to 20,000 parts per salinity zone expands, well then the geographic range, predicted geographic range expands, but they haven't really changed their preferred salinity range. Next slide. This is the early juvenile spot, which, as I said before, is a transient species. In other words, they move out into the Gulf of Mexico to spawn. They recruit into the Charlotte Harbor, in the northern part of Charlotte Harbor, and into the lower parts of the Mayaca and lower Peace River in the winter. But then in spring, they move north and they move up into the what's called the upper Peace segment, the upper part of the lower Peace River, north of where it intersects with Shell Creek. And so the question is, why are they doing that? Because you've got this happening with spot in the spring and red drum in the winter. They're moving up into this zone, and then in the following season, especially in this map, it's a little hard to see on my screen, but they've moved. There's still some spot in that segment north of the intersection with Shell Creek, but the majority of the population has moved down towards the mouth of Charlotte Harbor. So you have a series of blue spots, which is the optimum zone, near the mouth of Charlotte Harbor. So that's kind of surprising, first of all, because that movement occurred when these animals were about 6 months of age, whereas most of the literature, like in Louisiana, would indicate that they don't move down or move out into the Gulf of Mexico until sometime in the winter. So they're doing it earlier than in Louisiana anyways. And then the fall map, again, shows a reduction in the blue map, and that shows that they're moving out of the estuary. And so the blue spots, the optimum zone is diminished as they move out into the Gulf. Next slide. Can you go back one? Go back 2, please. Back. Can you go back 2, please?
Unfortunately, Peter, we are out of time, so we just wanted to have you wrap it up real quick. So we forwarded to your conclusions, and then we're going to switch over to Q&A.
Okay. Well, like I said, the water management district has been trying to manage for a decrease, a 15% decrease of salinities less than 2 parts per thousand. Despite rather large water withdrawals in summer, the predicted salinity grids and seasonal temperature grids within Charlotte Harbor did not differ substantially between baseline and minimum flows. Since the other variables, depth, bottom type, dissolved oxygen, were kept constant, the similarities of the salinity and temperature grids largely explain why seasonal habitat suitability maps were so similar for species life stages associated with baseline and minimum flows. The population numbers estimates, which I didn't get to show you, there's huge numbers of bay anchovy, for instance, indicate that the percent of flow reductions used by swift mud do not markedly affect the fish and invertebrate biological resources in the study area. So I'll take questions.
Thank you so much. That was a very interesting presentation, and I know we have time for just a question or two, about 2 minutes here, Peter, so feel free to answer as much as you can in that time frame.
Well, the first question I see here is, how does public weigh in to support diversion limit? I think the swift mud is, as has been shown in some of the other talks, is a member of many committees that have public input. And my agency also, of course, deals with the public. So I think one of the advantages of this study is it gives you a solid scientific basis for predicting what the influence would be of water withdrawals. And it's easy using maps to explain to the public what's going on. Second question is, are 2 different hydrodynamic models, DOM and inshore, used for this study? I have done modeling on the West Florida shelf, not on the same species, but that's possible. We have done habitat suitability modeling on the West Florida shelf. So it is possible to do them outside of the estuaries, if that's what the question is. Third question is, were all fish species trends examined or particular kinds of indicator species? I'm not quite sure I understand the question.
Well, that's great.
We couldn't look at all the species, so we had to select species that we thought were sensitive to freshwater inflow. Thank you.
All right. Well, wonderful. And if there are further questions, please feel free to reach out to Peter directly with his contact information in the proceedings document that's on the CHNEP.org website for today's event. Thanks again. And with that, we're going to move on to our last lightning talk, which is actually going to be delivered by me, Jennifer Hecker, with the Coastal and Heartland National Estuary Partnership, talking about the South Lee County Watershed Initiative Hydrologic Restoration Project. Next slide, please. So I'm going to go through this somewhat rapidly.